noir plasma chrome bleed. Unlocking knowledge, inspiring innovation, empowering curious minds.
Like what you shares.
By comparing the angiosperm xylem and phloem on this side with the human vascular system here, we can observe key evolutionary adaptations for nutrient transport.
In flowering plants, placentation is defined as the specific spatial configuration and attachment of ovules within the ovarian cavity. The anatomical categorization of plant placentation is determined by carpel number, the presence or absence of internal septa, and the specific locus of ovule attachment. Superficial placentation. This pattern occurs in multicarpellary, multilocular syncarpous gynosia. Instead of restricting in humans, the placenta is a temporary organ of chimeric origin, arising from the developmental integration of the... As you can see, the anterior placenta is positioned along the front wall of the uterus, acting as a cushion to buffer external pressure.
Here's the heartbeat, strong and steady. Everything sounds perfect.
All right, everything looks great. The placenta is positioned posteriorly, right where we want it to be.
As we zoom in, the cross-section clearly shows the placenta implanted at the fundus, which is the top of the uterus.
Lateral placenta. The placenta implants on the left or right lateral walls of the uterine cavity. This lateral asymmetry.
As we zoom in, the visualization demonstrates the pathological condition of placenta previa, where the fully developed placenta completely covers the internal cervical os.
As you can see here on the ultrasound, the placenta has grown through the uterine wall, which confirms the diagnosis of placenta percreta.
Placentation parameter, angiosperm su, human su, origin of tissue, purely maternal sporophytic tissue su, chimeric tissue. Placentation parameters cover both angiosperms and humans. In plants, we look at basal and superficial types, while human placentation can be anterior, posterior, or placentation parameter. Angiosperm basal or superficial, human anterior, posterior, or previa. Basal placentation features, monocarpellary/syncarpous, unilocular.
While both involve attachment, the angiosperm basal placentation anchors the ovule at the very base. Now compare this to
the initiation of seed development is driven by double fertilization, which yields a diploid zygote and a triploid primary endosperm nucleus. The terminal cell of the asymmetrical zygotic division undergoes multiplanar mitotic divisions to form a spherical mass of cells.
The zygote undergoes a precise, genetically programmable sequence of developmental stages. Heart stage, the rapid...
The zygote undergoes a precise, genetically programmed sequence of developmental stages. Torpedo stage, the cotyledon...
The mature angiosperm seed consists of the embryo and the endosperm enclosed within a protective seed coat, or testa.
The external surface of the seed displays key anatomical landmarks. Hilum, a physical scar on the testa marking the point where the seed diffuses.
In humans, the zygote undergoes a series of rapid mitotic cleavage divisions within the fallopian tube, forming a solid 16-cell sphere known as the morula.
To initiate implantation, the blastocyst must first hatch from the glycoprotein-rich zona pellucida. The process of uterine implantation progresses through 3 distinct sequential phases. Apposition.
keratin. The syncytiotrophoblasts projects finger-like invasive pseudopodia that secrete metalloproteinases, degrading the
Gibberellins activate growth pathways in plants, while simultaneously, oxytocin initiates uterine contractions in mammals.
The syncytiotrophoblasts secretes human chorionic gonadotropin, which travels to the ovary. Upon binding to the corpus luteum, HCG initiates signaling.
The transition begins as PRB receptors diminish, replaced by inhibitory PRA receptors. This shift enables the
Simultaneously, rising estrogen levels increase myometrial sensitivity to oxytocin and prostaglandins.
The physiological dynamics of delivery are significantly altered by the topography of the placenta. In both anterior and posterior,
Placenta previa occurs when the placenta implants low, covering the cervix. This interface between the placenta and the decidua is critical.
Keep the suction ready right here. Suction ready. Hand me the forceps, please.
The arterial line shows a sudden drop here, consistent with significant blood loss.
We should initiate the massive transfusion protocol immediately.
Suction coming in.
Scalpel.
Thank you.
The directional guidance of this pollen tube toward the micropyle is mediated by precise chemotropic gradients. Once through the ZP, the sperm enters the paravitelline space and attaches to the oolemma. In mammals, this crucial adhesion step is mediated by a The oocyte undergoes a two-step block, a rapid depolarization followed by the release of intracellular calcium oscillations which stimulate the exocytosis of cortical granules.
Following karyogamy, the developmental paths of plants diverge sharply.
In contrast, human embryogenesis begins with symmetric holoblastic cleavage. The human zygote divides into two equal Only around day 4 does cellular differentiation initiate, as the blastomeres segregate into an outer epithelial trophoblast layer and an inner cell mass.
In plants, cytokinesis is centrifugal. After chromatid separation,
the vesicles coalesce at the center to form a transient membranous tubule network called the cell plate.
The chromosomal passenger complex, containing Aurora B kinase, localizes to the overlapping antiparallel non-kinetochore microtubules, bundling them together. The contractile ring is an actomyosin-based structure composed of thousands of parallel filamentous actin strands and bipolar myosin II motor proteins. This contraction exerts an inward pulling force, creating a visible indentation on the cell surface known as the cleavage furrow. The cleavage epigenetic programming and genomic imprinting. Genomic imprinting is an epigenetic phenomenon wherein genes are expressed in a parent-of-origin specific manner. Upon fertilization, these hypomethylated maternal alleles are transcriptionally active, whereas the corresponding paternal alleles remain silenced by maintaining cytosine methylation. Any alteration of this dosage ratio in interploidy crosses disrupts the expression of imprinted genes controlling endosperm cellularization. These marks are established de novo during gametogenesis. In spermatogenesis, paternal-specific methylation marks are laid down.
Pathological and structural barriers restrict hybridization between different genera. The morphological and genetic differences render cross-kingdom hybridization impossible.
The prezygotic barriers, mechanical and gametic isolation. At the prezygotic level, the gametes of plants and humans cannot recognize, bind, or undergo membrane fusion. Adhesion deficiencies. Once inside the paravitelline space, mammalian gamete membrane adhesion requires the interaction of oocyte Juno and sperm Izumo 1. Homologous chromosome pairing would be completely absent. During meiosis, the chromosomes would remain as univalents, unable to align at the metaphase plate. Even if a hybrid genome could be maintained within a single nucleus, the epigenetic regulatory frameworks of the two kingdoms are entirely incompatible. Human embryogenesis requires global-like demethylation, followed by tissue-specific de novo. In a hybrid embryo, the plant glycosylases would fail to recognize mammalian methylation sequences, while mammalian In a plant-human hybrid, a human transcription factor would fail to recognize plant promoter sequence, and a plant ribosomal subunit would be structurally unable to bind in translation initiation. A comparative analysis of plant and human reproductive biology reveals that while both kingdoms have evolved sophisticated mechanisms for embryonic nutrition and development,
like what you share.
This website makes learning so easy. I found all my resources here.
Noir Plasma Chrome Bleed. Unlocking knowledge, inspiring innovation, empowering curious minds.